Okay. Marks the 31st year of our existence, so that's cool. Our agenda tonight is as follows. I'll start with highlighting a few recent accomplishments, then turn things over to Tim Sisson to share some big news about upcoming land acquisitions. Thereafter, we will hear the keynote presentation from Dr. Kendra Cipollini, a professor of biology at Wilmington College. So that'll be outstanding to hear. But first, some news. At our Kirby Nature Center, we've completed 2 major projects and a third is underway. In December, we finished our Prairie Path, which is compliant with the Americans with Disabilities Act. And that's a 6-foot wide path that starts at the left of the barn and loops around a portion of the prairie and includes a natural seat retaining wall for part of the distance. Once the prairie is in bloom, I think it'll be a great site. I'm certainly looking forward to it. These improvements will provide visitors with beautiful views of the prairie, the hillsides, and the barn. The project was designed by Rachel Robinson Design and was constructed by Jeffrey Allen Corporation. If you'd like more information about the Prairie Path, check out the article by our Vice President, Mary Perkins, in the most recent edition of the newsletter, the winter edition. As Mary explains in the article, the Prairie Trail is the first step in our plan to create a nature playscape for kids. This larger project is something that Mary has been working on for a number of years now.
Hello.
Second up, a second completed capital project is a renovation of our barn.
Where are you?
Okay, so if you're not talking right now, I think everybody should be muted. Okay, let's proceed here. A second completed capital project is the renovation of our barn, performing some very necessary upgrades to address deferred maintenance. This project by John P. Tumlin and Sons included the installation of a new metal roof, new rafters, new beams to strengthen the roof, and other improvements, including repairs to the siding, which looks good, and the installation of new electrical fixtures and receptacles. So personally, I think the renovation looks great and was very much needed. Currently underway is our third major capital project to build a footbridge to extend our Creekside Trail at Kirby. The bridge will extend over a tributary of Muddy Creek that runs parallel to South Road. This bridge is being designed and constructed by volunteers from the Miami University student chapter of the nonprofit organization Engineers Without Borders. And it's being overseen by one of the professional mentors for the chapter, Mike Bruner. The bridge will be 80 feet long, 80, so that's quite long, with steel trusses and a wooden deck, which will be 3 feet wide. Both the design work and the labor, all the labor for the construction is being donated free of charge. Construction is expected to occur in the spring. So big thank you to the Miami Engineers Without Borders who are volunteering their time. And I'd also like to do a shout out of thanks to our trustees, Roger Auer and Tim Sisson, for the coordination of the project from our end. Funding for all 3 of those projects was through grants from 2 anonymous donors, which we are deeply grateful for their generous financial support and excited to put that money to good use. One more news item I'd like to share here is that many of you know the volunteers of our Land Stewardship Committee are incredibly active. I mean, incredibly active. And as my final news update, I'd like to highlight a recently completed habitat restoration project at Kirby, at the Kirby Nature Center, performed over the last few months. Under the leadership of Tim Sisson, in February, volunteers finished clearing the honeysuckle next to the road along Main Street at the Kirby Nature Preserve, remarkably improving the aesthetics of the area. And that was a culmination of effort by roughly a dozen volunteers, as I understand it, performing many such work projects there over the last few months. So not only beautifies the Kirby Nature Preserve with some really awesome curb appeal along the street there, but also helps, of course, native plants survive and thrive. So thanks, guys, for that effort. So moving on then, another important aspect of land stewardship is acquiring the land to be protected. And I can attest that this is a huge effort that can take years. So next up, Tim Sisson, our land stewardship chief, will talk about some exciting things going on with land acquisition. So Tim, over to you. And you're muted. You are muted, Tim. You're muted. So Tim, we can't hear you. You're muted. There you go.
I think I'm unmuted now.
Yeah.
Okay. Thanks for the great introduction. And I saw John, I think I saw Matt here too, who both helped a lot with clearing the honeysuckle along the road there. And just one more thing to add to that is our benefactor for the Kirby Nature Preserve was really interested in seeing it protected or seeing it cleared. And then also, to me anyway, it was just embarrassing that we say so much about clearing honeysuckle and how, boy, we have done such a great job clearing honeysuckle in our properties. But then people driving along our main nature preserve there in Addiston would see masses of honeysuckle along the road. So that just didn't seem like a good idea to me. So that was a main impetus for me to help organize that and get the stuff cleared out. Now I'm gonna talk about a couple of properties that we've been working on for years, and just all together within about a year, we will be protecting them and adding a third to the amount of land protected by organization. It's just amazing how these things all kind of come together. But we will be protecting a floodplain forest in Sailor Park that we first started on 6 years ago. John and I first met with the owner over there and started the dialogue, and we have come to a conclusion. We will definitely be buying that probably next year. And let me see if I can bring up the map of that. I know it's here. I just saw it. Yeah, here it is. So I should be sharing that map. Can you see it there?
Yeah.
Is it viewable by everybody? Yeah, okay. Well, in light blue here is the property. And this is Muddy Creek, the dark blue line. And we are just across the road from Kirby Nature Preserve. That building right there is the Kirby Nature Center. So we were right across the road there with this beautiful 11-acre floodplain forest, great big mature trees. That is going to be ours. Probably early next year. I mean, we have total agreement on the price and everything. So we will be buying that property. As part of the conditions for the purchase, we were required to plant over 100 trees there. And boy, we really did that. Debbie and John, Matt, were all part of that project where we planted between 130 and 140 trees there last fall. So I'm really looking forward to seeing how that looks. This is Sailor Park over here, just to help you a little more with the perspective. Gracely Drive here. So it's just right in our neighborhood, and we're really happy to see it there. It's the first floodplain forest we will be protecting. It'll have a lot of animals that we have never had before in our preserves. I saw a mother wood duck with 3 chicks swimming right here last spring. We've seen snapping turtles there. We've seen great blue herons. So it's really a great property. I'm just really happy after all this time that we'll be able to protect that. So let me go back now. The next one that we will probably be closing on March 22nd. We again started working on that 7 years ago, started negotiating on that property, and we came to an agreement on price last year. We were able to apply and receive a grant from the state of Ohio for about 70, well, not about, but 74% of the purchase price. And Kendra was really instrumental in helping us with that. I'll get to that later when I introduce her. So that property, if I go again here, is on Rapid Run Road. Yeah, this is Rapid Run Road coming down the hill. This is Hillside Avenue. So it's this big chunk of mature forest. Again, about 11 acres on the side of the road. We currently own this property and this property. We own this property here. These are all part of the Whitetail Woods Preserve. And we also own property over here, which is a Buckeye Trace Preserve. So it's going to almost link those two big preserves and have over a mile of hillside protected from almost Bender Road way past Rapid Run, which is what we're all about. I mean, Western Wildlife Corridor means we want to set up protected wooded corridors along the Ohio River. And this is a perfect example of protecting the hillsides for, gee, it'll be over a mile once we optimistically, once we protect the intervening properties here. So that's those two. We will close on that in December. What am I saying? On March 22nd. So the third one, I'm not at liberty to say where it is at this point. It's confidential at this point. It's a very large property that's very close to an existing preserve that we own. Again, mature forest on the slope facing the Ohio River. We first started talking to the owners of that property over 20 years ago about somehow protecting it, and we've worked it out. We will be protecting it later this year. So I am really happy about that one. So we are moving along, really rapidly toward actually protecting the corridor, which is so satisfying after all this time. So now let me get to our presenter, Kendra. Back about, it was almost a year ago when we were looking at the Rapid Run property and the Muddy Creek property. We knew that we wanted to apply for a Clean Ohio grant to purchase them. Clean Ohio grant is grants provided by the state of Ohio for green space. They will provide up to 75% of the purchase price and other direct costs. And we really wanted that because that'll save us upwards of $100,000 on those two properties. So they are very complicated applications to complete, to get all the materials together, you have to be extremely organized. So we need somebody to help us with that. So I reached out to other land trusts, other organizations in the region, and the people over at the Tecumseh Land Trust in Yellow Springs, who I knew, they recommended Kendra. So I contacted her. We exchanged emails and phone calls, and came to an agreement. Yes, she would help us with those grant applications, which just worked out incredibly well. Yeah, Kendra, I can't thank you too much. I can't thank you too much. That is so much work. And by sticking to it, by being so organized, you were able to get all the materials together, all of the information submitted in a timely manner. So that, as I said, we will be closing, it looks like, on March 22nd, to purchase those properties. We will getting, correct me, isn't it going to be around $270,000 from the state? It seems like it's, I think it's in that neighborhood.
About that much, 265 or something like that.
Right. A huge chunk of money. Boy, for our organization that survives on donations and memberships, $270,000 is a big deal. And we will be getting that from the state, and we'll be closing on that property in March. So that's my property wind up, and going on to more on Kendra. She currently is a professor at Wilmington College. She has worked for the Cincinnati for the Nature Conservancy as a director of science. She worked for the U.S. EPA as an ecologist. And I guess it was before that you worked in wetland delineation for engineering firms, Kendra. I mean, really, really an impressive resume there of her her past activities. She currently does research on invasive species, endangered species, and conservation genetics. And we've already talked about invasive species control over lunch at Cabana. And I'm hoping that we will have more conversations about that in the future as we deal with many invasive species on our properties. So that's what I have. So with that, Kendra, I will turn it over to you for your presentation.
Okay, thanks. I'm going to go ahead and share my screen. You gave me permission, didn't you? Okay. All right. So I wanted to also let everybody know that if you have a question as I'm talking about something, feel free to interrupt. You can just speak up and interrupt, and I'll answer your question. Somebody else does too. So as we go along, if you have questions, please let me know. Okay, sorry, it said Zoom quit unexpectedly. You're there though, right? Okay, I just got a warning on my system. Okay, I just got it again. Just gonna
You're sharing, everything looks fine. You're sharing your main screen, which everything looks fine from my point of view.
Good, too.
We just lost her.
Yeah.
well, I'm here now again.
Yeah, just as I said that, everything went back to the checkerboard.
I know, it told me twice. There's been a problem. Okay, let's try this again. Okay, so I did change the topic slightly. I don't know who's in charge of the Zoom, but there are a person or two that are still unmuted. So you can just mute them from your desktop so we won't hear things. Okay, so I did change the topic somewhat. I am talking about riparian ecosystems. I was asked to give a talk about riparian ecosystems, and I thought I'd give a talk about wetlands. But as I was thinking, I thought I would revise it and talk about invasive species in riparian habitats, because that's where most of my research lies. So I've switched it a little bit, but I promise it's still informative. So thinking about river ecosystems, there's something called the river continuum concept. And it basically describes how a river changes from the headwaters down to higher orders as it gets bigger, essentially. Okay, and so how does the community change, and how do ecosystem dynamics change as you go from headwaters to downstream? And one thing I wanted to point out, and I didn't really know this for a long time or really think about it, but you know, what do organisms up in the very, very headwaters of a stream eat? Where do they get food from? Right? You might go, oh, they're going to eat insects. Okay, well, where do those insects get their food? Okay, I didn't know this for a while, but the major food source for headwater streams is something called coarse particulate organic matter, which is right there, CPOM. You guys there? Okay, that was just somebody breaking in, I guess. In other words, leaves. Okay, the major food source for the head of the stream is coarse particulate organic matter, which is essentially just pieces of leaves. So that forms the basis. So we're still talking about terrestrial ecosystems forming the basis of the food web for aquatic ecosystems. And so when you look at how animal communities change as you move down the stream, when we look at the top of the stream up here, these are the types of species that you see. Okay, you see things that break down big leaves. You see shredders. You see some other things that break down leaves. I've got another figure here in a second, okay? But one thing I wanted to mention is that coarse particulate organic matter becomes fine particulate organic matter as it moves downstream. And you can see here that fine particulate matter is still really important. And it's still really important down here, even in large rivers. So it's still the things that fall into the streams that provide a huge portion of the basis of the food web in stream ecosystems. Sure, there's some plants growing, there's some producers, there's some phytoplankton that form a basis of the food web. There's some algae. Here's some algae vascular plants, okay? But a lot of the major input into streams that forms the basis of the food web is plants, terrestrial plants. Okay, so here's that coarse particulate organic matter. This is a food web in a headwater stream. It falls in the river and it's broken down. By microbes will break it down. It will dissolve and become dissolved organic matter. It becomes fine particulate organic matter when things like shredders shred it up. Okay, and then it becomes smaller. Once it becomes small enough, then collectors can eat it. Collectors use things like nets or filter, they're sort of like filter feeders. They'll filter out these fine particles, where shredders eat more of like the leaves, they shred them up, hence the name. Okay, there are some producers in headwater systems. There are some plants that live like on rocks, phytoplankton, etc., that are important to the basis of the food web. But the really important thing is right here, plants. And that's what I study. Okay, and and those plants, so here's the major system. This is a headwater ecosystem. I just wanted to point out here that those plants affect the insects. The insects then affect what eats them, even back on the land again. So it links back to terrestrial ecosystems. So if you don't have enough leaves going in, you're not going to have enough insects. You're not going to have insects emerging. You're going to have less birds, bats, lizards, spiders, right? So having a lot of leaf fall come in is important. And also having the right kind of leaf fall coming in is important. So here's kind of my thus, summarizing those two slides. Terrestrial plants are the basis of food web for a lot of riverine systems. They provide the base food that everything else shreds up and eats. Okay. If you modify habitats along these streams, you will affect ecosystems all along the river. Okay, and the quality of the riparian habitat and also the quantity. So how many leaves are coming in, also the quality of the leaves coming in. So that's sort of what I'm going to go to next, is some of my research looking at what some different species that are found in riparian habitats and how they might affect water quality. Okay, so invasive species is an example of something that modifies riparian habitats. And these are 3 invasive species that I work with and have done a lot of research with, and I am sure you're familiar with all of them. Okay. I like this picture, but because it has all 3. This is taken at Lytle Creek in Wilmington, where my college is, and there's garlic mustard here in the front, there's celandine here in the middle, and our old friend Amber honeysuckle here in the back. Okay. All of these are going to be important inputs in the stream. So understanding how they work and what their chemical composition is important to if they're a good food source in rivers. Okay, so my research mostly focuses on the plants. So I don't really work with rivers, but I work with plants along rivers, and that has an implication for what's going into the rivers. So I'm going to talk about some of my research that I've done with these 3 species, looking at their impacts on riparian systems. And then at the end, I'll talk about how those impacts might lead to issues in the river itself. Okay. So just a little background. I know you guys are all familiar with it, but Linosauromachia, we're kind of at ground zero of Linosauromachia. This is the location where it was introduced. And as you know, it takes over woodlots, but you can see it's pretty well distributed across a lot of the United States. Everything in green in that map is where it's found. Ranunculus ficaria is a fairly new-ish invasive species. It's really been like the last 15 years or so. Amber honeysuckle we've known about for 30 years. And it is found in the Northeast, Midwest, and then there's a few populations over in the Pacific Northwest. And that is a different variety, actually. We have a variety here called Ranunculus ficaria variety bulbosa. Okay, that's a slightly different variety than the one that's found, but that was a horticultural species that was released, and Tim did talk about that release in Cincinnati. And this is a picture, actually, this is where I did the study, and that is Hamilton County Parks, Sharon Woods. Couldn't remember which one. That's Sharon Woods there, that picture. The last species that I deal with a lot is garlic mustard. This is also a fairly old invader. It's actually probably becoming less important. It is starting to die out in a lot of woodlots, so it's probably not as big of a deal as it once was. We're still studying that, but you can see it's also distributed across much of the US, and it was introduced from Europe. It's interesting because I had a friend that went to do they went to the UK to do a tour, and they went to a greenhouse, and one of the nature preserves was propagating garlic mustard to put out in the field because there it was important. And they were like, I can't believe I'm seeing them growing this thing, but they are. And I have gone to Europe to see garlic mustard in its native habitat in France and Ireland, and it's just small amounts there. So a couple of my research projects, I'm going to go through a couple of my papers and try to highlight some of the relevant aspects.
Excuse me, do they
Yes.
know why garlic mustard is dying out? Is it on its own? It's life cycled out or what?
Yeah, they're trying to figure that out. They just think that it's sort of had this competitive advantage that now it no longer has. They're not, I don't know that they know a mechanism for it, to tell you the truth. I just know that they're thinking that it might have this kind of cycle where it like flushes and it's really prevalent for a few years and then kind of comes back and then kind of flushes and then kind of goes down, but that it doesn't maintain these really, really dense patches for a long time period. So I don't know if it's like competition, intra-specific competition. I don't know if it's, you know, soil or environmental conditions. I'm not sure what it is, but they
Okay.
are finding this sort of like flushing, going away and then moving to a new area, etc. So yeah.
Thank you.
So my first question, this is a question, I wanted to look at amber honeysuckle and understand if it's allelopathic. I will talk about that. And does its effect vary with different nutrients? The concept of allelopathy you're probably familiar with. It is the fact that plants will release chemicals from their leaves, from their roots, that then affect the growth of another plant. That's called allelopathy. And those chemicals that they release are called allelochemicals. Okay, and one that you're probably familiar with, the poster child for allelopathic species, is black walnut. If you have a black walnut, you know you cannot plant much under it, it dies. And it's not due to the fact that there's a lot of shade under black walnut. It's due to the fact that black walnut produces something called juglone. That's the allelochemical that inhibits the growth of other plants. So if you go to plant your impatiens under your walnut tree, they don't do well because the allelochemicals kill it. So my question is, does honeysuckle have these allelopathic chemicals? And one way to study allelopathy is by making what we call teas. You essentially can take leaves of the species, like you see here. This is honeysuckle right here. You can take honeysuckle leaves, weigh them to a certain mass, soak them in distilled water for about 48 hours, filter out the leaves, and then you end up with this delicious looking tea, which has chemicals leached from the leaves into that tea. You can then take that solution and add it to other plants. So in this study, we just used different concentrations of Lonicera macchii, that's amber honeysuckle, different concentration of amber honeysuckle teas to see how it impacts the growth of a plant called Arabidopsis thaliana. That's the picture here. This is the model organism for plant biology. It's like fruit flies are to genetics. Arabidopsis is to plant biology. So I just use this sort of common plant to see, does honeysuckle affect its growth? Okay, and so I had 3 different levels of honeysuckle extracts. Here's the control, grown with no extracts. Here's low, and here's high concentration. Okay, and then I looked at things like growth and reproduction. I'm going to focus on the bottom, which is reproduction. The salique is this little structure here. It's the reproductive structure on mustards. It's called a salique. That thing on the garlic mustard, that seed head on the garlic mustard, those are called saliques too. Okay, so if we just focus, so if I look at high concentrations of nutrient, these are 3 different nutrients: low, medium, high. I'm just going to focus on high. At low extract, I made 120 saliques. At the control, I made 120 saliques. And you can see it went down to 1/3 of that when I added high extract. So we see a general decline in seed production with honeysuckle extract. That's telling me that adding that extract to the soil negatively affects growth, which means that honeysuckle is allelopathic. I don't see a big decline at low nutrient levels. Medium nutrient levels, it goes down a little bit. There's just a bigger effect at high nutrient levels. And so the major take-home conclusions from that study is that amber honeysuckle leaves are allelopathic. They create chemicals that inhibit the growth of other plants. It was affected by nutrient conditions, but the main point for this talk is that honeysuckle is indeed allelopathic. Okay. My next research question was looking at amber honeysuckle and garlic mustard and figuring out, okay, we know that they harm other plants. We're pretty sure they do. Actually, there was a dearth of studies that actually showed they harm other species. It was anecdotal, but there's some. But anyway, how do they affect it? So when you see honeysuckle in the field, is it affecting plants that grow underneath it because of those allele that I just talked about? Or is it affecting the plants underneath it because of just shading? Right? If you just shade something as heavily as honeysuckle shade things, that will affect it. So I wanted to figure out.
Kendra,
Yeah.
I did have a question. Would the chemical, chemical that they produce, would that be a general, have a general effect on all plants, or would it more specifically affect some plants more than other plants?
Well, now you're getting to the end of my talk.
Yeah.
It does
Sorry.
depend. That's a great question, because yes, it's not universal. It doesn't affect all species the same. Some species are more resistant, and some things like garlic mustard sometimes works to kill the mycorrhizae, and then that hurts the plant. Some species like amber honeysuckle doesn't seem to work through mycorrhizae. It seems to work directly through. Garlic mustard might be more of a mycorrhizal disruptor. So it has different mechanisms, but that's a great question. So I wanted to ask, how do garlic mustard and honeysuckle affect native species in the field? What are they doing? And the 2 things that I was interested in was allelopathy, okay? And one way that you can manipulate allelopathy in field soils is you can add activated carbon to the soil. Activated carbon is the same stuff that you buy at the fish store to, take all the contaminants out of your fish water. You can buy the activated carbon at the fish store, mix it in with the soil, and any allele in there adhere to the carbon, and you can essentially take the allele out of the soil. So it's a way to manipulate allelopathy in the field. It's not perfect. There's some issues, but it's one thing that people use. So I wanted to know, is allelopathy occurring in the field? The other thing I wanted to know is above ground competition important? So is it just light blocking, which I thought it might be for honeysuckle, okay? So I designed an experiment to manipulate those 2 factors. I had and I used areas with garlic mustard and areas with amber honeysuckle, okay? And what I did is I cut, in some areas I cut the invasive species. So I cut the garlic mustard down, or I cut the amber honeysuckle down. And then I left there as a control. I had a control where I didn't cut it. And then I also added carbon to the soil to manipulate allelopathy. So these treatments here would have no. Okay. And then I have all the combinations of them. So I have some plots that have carbon added, but the plant's not cut. This one, carbon is added and the plant is cut. So I'm getting rid of both allelopathy and above ground. For this one, I'm just getting rid of allelopathy, but not above ground effects. This one, I'm getting rid of neither. This would be my true control. This is like, I don't do anything. But this one, I'm getting rid of, I'm keeping the allelopathy in place because I'm not adding carbon, but I'm cutting it. Okay, so I'm getting rid of above ground. So by looking at all those combinations, I can get at, is it above ground? Is it allelopathy? And then I did it for 2 species and I replicated it. And the species, so what is the native species that I used? The native species that I used was Impatiens capensis or touch-me-not. And I picked it because it's found in the same habitat as Lonicera macchiae, as amber honeysuckle and garlic mustard. And it's also an annual, so I can measure reproduction on it. So what I did is in these plots, these treatments that I just talked about, I put in 6, I planted in 6 impatiens plants in each plot. Okay, and then over the course of time, over the course of the field season, so I went from, seedling to seed. So it was, 2.5 months. I measured every 2 weeks, I measured height of the impatiens. I measured the number of fruits. I measured survival. Okay. And there, of course, are those little fruits that you can't touch on the touch-me-not. So I wanted to know, how do garlic mustard and honeysuckle, how do garlic mustard and honeysuckle affect touch-me-nots? So that was my target native plant. Okay, so these are some of the results. Okay, from that, and I'm going to just point out some of them. Okay, so one thing that happened is that these over here are the cut treatments. These are the uncut treatments. And the major effect I had here was cutting. If I cut the honeysuckle, this is the cut one, it lived longer. It died sooner if I didn't cut it. This average is here. Same thing with garlic mustard. If I cut it, I didn't cut it. Yeah, if I cut it, it lived longer. If I didn't cut it, it lived a shorter time period. So above ground competition is important. I can see it's more important in Lonicera because look at that big difference. It lasted a lot longer when I cut it. It died much more quickly if I didn't cut the above ground on Lonicera. Okay, but I did also find evidence of allelopathy. Okay, particularly in garlic mustard. So these are the garlic mustard plots. And you can see that this is, we can look at number of fruits here. You guys can still hear me, right? Because I hear that like humming and my computer just dimmed. You're still there, right?
We can hear you.
Okay. Okay, my computer
can
just
hear
did something
you,
weird and I
Kendra.
heard this little mumbly thing. So I wanted to make sure you were there. All right. So I'm going to focus on the one on the bottom, which is the number of fruits. And you can see that, first of all, this is the cut. It's much higher than the uncut. So it produces a lot more fruits when you cut it. This is garlic mustard. But carbon had an impact. So these are both not cut. So the garlic mustard is still there. In this case, there is allelopathy, and here there is not allelopathy. So you can see that I was able to increase the amount of fruits without cutting the vegetation, but getting rid of the allelopathy, putting in the carbon. So if I put in the carbon, I take the chemicals out, and I can get production pretty much as high as if I cut it. Okay, so that's pointing towards the fact that allelopathy is an important mechanism in the field. This is amber honeysuckle. And I didn't see big effects on reproduction, and I did not see that carbon effect. So allelopathy, at least in the field, in this study, did not matter a ton for amber honeysuckle. For amber honeysuckle, the cutting was much more important. Okay, you can see these are the cut, these are the uncut. It's much higher.
Yeah.
Okay, so that's the conclusion from that. both above ground competition and allelopathy are important in garlic mustard. So I am seeing allelopathy in the field in garlic mustard. I'm not seeing allelopathy by honeysuckle in the field, but I am seeing that shading effect. I was able to document that effect. Okay, and that's in this paper. I put my papers at the end. I guess I forgot on that first one to put the paper. Okay, the next species that I wanted to study was Ranunculus vicaria, and it was lesser celandine. Sorry, I'm trying to go with common names, so to be consistent. But lesser celandine, when I started to study it, this study was done in like 2010. And when I started to study it, you know, you see, this is again, Sharon Woods, and it was growing like this in Sharon Woods, and you see it like that taking over, and you're like, man, that can't be good, right? So I go into the literature and I try to find something that shows what it does to native species, and I can't find any papers on it. It was banned in Connecticut, but nobody even had any evidence that it did anything to native plants. Now, it's kind of a no-brainer. When you look at something like this that takes over, you're like, that can't be good. Let's go ahead and ban it from horticulture. But you know, it was declared a noxious weed in some states, and we really didn't have any evidence that it was bad. So I said, okay, I'm going to set out to develop a study to show that it's having an impact on native species. Lesser celandine is also called pilewort, Pilewort, W-O-R-T, pilewort, and it's used in herbal medicine. And it goes back to the days of when what the plant looks like is what it treats. So for example, ginseng, the reason why it's supposed to be so good for you is because if you pull up a ginseng root, it looks like a little man. And so since it looks like a little man, it must be good for the whole body. Okay. They thought that the roots of lesser celandine looked like hemorrhoids. And so they thought it's good for piles or hemorrhoids. Okay. And so that you'll see it sold for hemorrhoids because the roots look like hemorrhoids to someone. And so when you see something that has herbal use, that usually tells you it's got some kind of chemical in it, perhaps, that people are using. And so that told me that lesser celandine might also be allelopathic. So I wanted to document, does it have an impact on the native species, and is allelopathy important? Okay, here's a picture. Again, you guys know about this. This plant is a pain, especially this variety bulbosa. It's called variety bulbosa because it makes these little bulbules that float downstream, and then each of these bulbules can germinate into a brand new plant. Here's the roots that they thought looked like hemorrhoids. And so they used it to treat hemorrhoids, not orally, because it is poisonous to take. So don't get any ideas. I think you have to actually insert it that's the method of use for it. And I want to know, did it have an effect on native plants? Okay, so here's, trout lily and violets in early spring. Again, this plant comes out very early, and so it really overlaps with our spring ephemerals. And our spring ephemerals have escaped a lot of competition by coming out very early in the season. Okay, so they come out early, but unfortunately ranunculus does too, right? It's probably out now. Usually comes out, even in a good year, it comes out mid-February. And then it's usually gone by mid-May. And so it's this sort of window in the spring that overlaps exactly when all those spring ephemerals that we value so much in our woodlands are out as well. So I wanted to know, does it have an impact on the native species? So there's my questions. How is it affecting those native species? Is it nutrient competition, or is it allelopathy? So I set up a series of experiments where I manipulated three things. I manipulated if ranunculus was present or not. So I had some plots where it was present, some plots I removed it. I had some plots where I added fertilizer and some where I didn't. And I used that activated carbon again to manipulate allelopathy in the field, and had some plots with activated carbon and some plots without. Okay, and that's a fully factorial experiment. What I mean by that is that I had all treatment combinations. So I had plus ranunculus, plus fertilizer, plus carbon. I had plus ranunculus, minus fertilizer, minus carbon, right? All the different combinations you can have of those three factors together. And then it was replicated. I did this in Sharon Woods, again, this is a picture of my field site. Okay, you can see I chose an area that was a nice, beautiful carpet of Ranunculus ficaria, and then I put my treatments in there. And then measured it over the course of the field season from May to August 2019. These are the plots. So these are about 10 by 10 centimeters plots where I removed ranunculus, I added carbon, I added osmocote, nutrients, etc. And then I planted in my old friend touch-me-nots again. Again, touch-me-nots make a good species to use. Number one, because nobody really cares if you move them. They're not like a highly endangered species. So I dug them up nearby and retranslocated them. And then also they're annual. So I can go from seedling to seed. So I can look at reproduction. If I were to look at something like wild ginger or violets, because they're perennial, it's just harder to get an idea, because one field season might not affect it that much. So I wanted to use an annual. So here's an example, and this treatment is minus ranunculus, minus carbon, plus nitrogen nutrients.
And
Okay.
why did you add the fertilizer?
Well, I wanted to know, like, are they just competing for nutrients? So is ranunculus beating out the impatiens just because it's taking away all the food, underground resources? So if you add nutrients in, they should be able to coexist together if they're competing for nutrients.
Okay.
Yeah, because I wanted to know that, because, you know, they could just be competing for nutrients, and that's what's doing it. So that's another one. The other reason I use nutrients is because carbon, it turns out, adds a slight amount of nutrients to the soil. So I'm kind of controlled for that as well. I told you there's some issues with using carbon as a manipulative tool. So adding nutrients can help get rid of some of those little nutrient effects. There's very few nutrients in activated carbon, but there is a little bit. Okay, and so these are just some results from them. So number one, I saw that survival of impatiens tended to be reduced when there was lesser celandine. So this is lifespan. So if lesser celandine was present, they lived about 90 days. If it was absent, they lived over 100 days. So they died 10 days earlier. Okay, I saw that it did affect fruit production. So this is number of seeds. We had to go out and try and capture all the seeds before they exploded from the plants, which we went out and did on a pretty regular basis. So this is no carbon. This is a natural condition. And if I have no carbon, whoops.
As you know, the screen disappeared, your slideshow disappeared again. So reload it.
I don't hear her either.
Maybe she disappeared too.
Give her a moment to reconnect.
The invasive plants did not like what she was saying, and they probably surrounded the house and got her during their talk.
Yeah.
I like that, Bob. Good point.
Someone just,
I felt them coming after me once in a while in the field, Bob, by the way.
I wanted to hear, someone just told me that they think they heard lesser celandine dies out after 5 to 10 years because it's taken the nutrients in the soil. I don't know what that leaves in the soil, but someone just told me that.
We can ask our speaker that when she comes back.
Hope she does.
I'm gonna try to text her, make sure she knows that she's been disconnected.
Is anyone using any?
I'm back.
Oh, there she is.
I'm sorry. My computer shut down.
Welcome
You know why my
back.
light dimmed before? My computer was not plugged in, so it just shut down. But we're back. All right. So I was right here talking about reproduction. So these are no carbon. So there's allelopathy present. These are with carbon. So I have gotten rid of allelopathy. You can see if I do not get rid of allelopathy, when ranunculus is present, it's much lower than when it's absent. See how much seed production is reduced by the ranunculus being present. Okay. However, if I add carbon, I can rescue that. Now my pen's not working. I end up rescuing it. Look, I get just as much made whether ranunculus is present, this is ranunculus present or absent. Here, when it's present, it reduces it greatly. So that's telling me that the carbon is ameliorating allelopathy. Okay, so I am seeing allelopathy in the field. I'm not going to talk about my nutrient results because they're not really important to this. I'll point you to my paper if you want the whole gory story. But conclusion from that study, the presence of lesser celandine does negatively impact a native target species, touch me not, and it does exhibit allelopathy in the field. So I'm showing it does negatively impact and does have allelochemicals. And that's in this paper that was put out in 2011. Again, that was done in Sharon Woods. The last study I'm going to talk about is some seed germination studies that I did. So we know that we know from other studies that we know from my study that garlic mustard is allelopathic. We actually know from other studies that honeysuckle is, though mine didn't quite show it. We know ranunculus is allelopathic. Which is worse? Like if you're going to choose to, you can ameliorate allelopathy, for example, perhaps by putting charcoal out in the field. That might get rid of allelopathy for you. So, which species should I control? Where should I put carbon out? How do I prioritize? So that was my question here is, if I look at garlic mustard, honeysuckle, and lesser celandine, what's worse? Okay, how is their relative strength of effect? Then does it vary with the species you're testing it on, and also whether you're looking at roots or shoots? Okay, so most of the other studies that I talked about, that some of the tea studies were done with shoots, but what's it look like with roots? So I again used those teas and I looked at ranunculus, or looked at lesser celandine and some of the other ones. This is comparing the effect of extracts of garlic mustard, honeysuckle, and lesser celandine on growth of that model plant, Arabidopsis thaliana. Again, there's a clip of the leaf. And how does it affect reproduction? And I can see that Lonicera macchii and lesser celandine significantly reduce it. They have a less impact than garlic mustard. Now I expected that because it turns out garlic mustard and Arabidopsis thaliana are in the same family. So the chemicals that garlic mustard has are similar to chemicals that Arabidopsis has. So the Arabidopsis is not affected by garlic mustard because they have similar chemistries because they're in the same family. Okay, so the garlic mustard is sort of a control, but I can see that Amur honeysuckle and lesser celandine have an impact on growth of this. I also did seed germination studies. Okay, very simple. You just wet a paper towel and you put seeds on. This is a funny aside. When I didn't have any pictures of my seed setup, so I Googled seeds on wet paper towels, seed germination on paper towels, because it's a fairly common technique. And all the pictures that came up were pictures of people germinating marijuana on wet paper towels. So that's what this is a picture of. But I did it with other seeds, but you could do it with marijuana. I could test this with marijuana, but I did not use it. The species that I used, I did the first set of experiments I just used common agricultural species, cabbage, basil, and leaf lettuce. Cabbage is in the same family as garlic mustard, so I expect cabbage will not be affected by garlic mustard. And so I added the teas, those extracts, to the paper towels, and I had a low, medium, and high. This is low, medium, high concentrations of extracts. And then I had 3 different species. So for example, this across the top is Amur honeysuckle. So how many seeds germinated with amber honeysuckle? You can see it had higher germination than lesser celandine. And you can see in this case, garlic mustard. So garlic mustard had the biggest impact on basil. But for cabbage, which is at the top, amber honeysuckle had the biggest impact. Okay, so the impact varies from species to species depending on which you're testing it on. I also did another experiment where I had native species this time. I use this tall thimble because it's in the same family as lesser celandine. I also use bottlebrush grass, hairy woodmint. These are all fairly easy to obtain and they germinate fairly readily. Some of the native species take a really long cold period, so these are ones that germinated pretty readily. Then I looked at leaf and root extracts of those 3 species I just talked about. So I had root extracts of garlic mustard, root extracts of ranunculus, leaf extracts, etc. Because I wanted to know, is it the leaf or the root? Okay, so these are the 3 species that I used. Again, lesser celandine, amber honeysuckle, and garlic mustard. And these are the 3 different test species. That's the mint. This is the thimbleweed. This is the bottlebrush grass. This green line is where the control with no extract germinated. So on my control had about 7 seeds. So you can see that for the root extract, it didn't have any impact on the bottlebrush grass. The main species that had impact here was this one, reduced germination, garlic mustard. So garlic mustard had the biggest impact on that. Garlic mustard also had a big impact here, but so did the other ones. These are all below that control line. Okay, so one important thing you'll see here is that the roots, which are the dark ones, are always higher than the shoots. So shoots had a much bigger impact than roots did. So shoots are more allelopathic than roots, and they're all allelopathic, but the effect varies with the target species you're looking at. So that's the question that Tim asked at the very beginning. It does depend which species you're talking about. And these are the 2 papers for those studies. Okay, so to tie back to river systems, since that's where I started, those allelochemicals that are negatively affecting other plants also negatively affect insects that try to eat them. Okay, so when these leaves are put into stream systems and shredders try to eat honeysuckle leaves, they do not do as well as if they're eating native leaves. And there's some people at University of Dayton that have looked at that. They've looked at honeysuckle streams versus non-honeysuckle streams, and they find that the community compositions are much different between them due to that. You can also do laboratory experiments where they feed different amounts of the leaf to the aquatic insects and see how they do. And they've seen that some of these plants do negatively affect. There's some studies looking at Phragmites and frog populations, for example. The Phragmites being in the wetland affects frog reproduction, okay? Because frog tadpoles are herbivores and they need algal communities, and you don't have good algal communities if you've got allelochemicals from the Phragmites in the water, right? It's a whole cascade effect. So the input, so the type of species that are going in there can affect the aquatic communities. Now, I've shown that all of these allelochemicals are bad to other plants. They have allelochemicals. The next step is to find out what they are. We know what some of them are in some of the plants, but then also to find out if they have effects on aquatic systems. We do have preliminary evidence that they do. Not for ranunculus, though. Nobody's done anything with ranunculus that I know of, or garlic mustard in aquatic systems, but honeysuckle definitely. The other issue is that a lot of native species can have allelochemicals or be difficult to digest. So, you know, think about oak leaves. Oak leaves have a lot of tannins. They're difficult to shred, but native species would presumably evolved tolerance to them because they've been exposed to oak leaves for a much longer time period than they've been exposed to honeysuckle leaves. So when you control those invasive species in riparian areas, it can have impacts on the entire river system because you're removing those potential novel allelochemical filled leaves from the system, and you're allowing the native leaves to go on, which of course would be what the river's used to. So, you know, protection and managing those riparian areas for native vegetation, getting rid of those invasives are really important to river health. Okay, so yeah, I went from rivers back on land, all my plant research, back to rivers again, hopefully tying in, convincing you that these species are not only bad for the plants underneath them, but they have real potential impacts on the river system as well. Okay, so that is the end, and I'm sorry about the technical difficulties, but I'm willing to take any questions.
I had a question in a kind of a different direction, but since you're in academia, maybe you would have some information that I haven't run across. But I know there are a lot of researchers, especially in Ohio State, I've heard, who are looking for some control from the country of origin, like Lanicera. They're looking for something from Eastern Asia that controls Lanicera, and to bring it over here and control it over here. Have they had any success with that? Of course, the big question is, would it hurt native plants over here at the same time? So do you know if they've made progress there?
I don't know that there's real big biocontrol efforts for Amur honeysuckle. I do know that Rick Boyce out of NKU, Northern Kentucky, he has done work. There's a virus that attacks honeysuckle that he's been finding. And so I don't know how far he's gone in identifying it or looking at effects, like does it really reduce it? I know my husband and I have done work on, there's a powdery mildew that affects garlic mustard. So we've looked at different questions related to that, if it could be a possible biocontrol, but I don't think, because you know, it's not just insects we could bring in, we can also bring in viruses and other pathogens, fungus. The good thing about this fungus for garlic mustard is that it's a native one that's already here. So you might just have to manipulate it to get it to grow better on garlic mustard in some way, manipulate conditions so it grows better, or just inoculate it really well. So, you know, there are some things that people are looking at for biocontrol. I know that there's something they're looking at for tree of heaven. It gets like this canker disease. And so they're trying to find that disease to see if it can be used for biocontrol. But none of that's very far developed. I mean, I think the best developed one, of course, is purple loosestrife, has the beetle that kills it. They've had good success with releasing beetles to kill purple loosestrife, but I don't see that. Do you guys see purple loosestrife down in Cincinnati? I don't see that too much. I see that more in the Northeast. I think
There's some around Cincinnati. I don't see very much of it.
Yeah, it's not like, if you go to the Northeast, like New York, Massachusetts, I'll see it a lot more. And even like Cleveland, I'll see it more often. We have it a little bit here, but yeah.
Yeah, we haven't found much of it in our preserves at all, just a few plants here and there. Well, thank you.
Yeah.
I've got several questions. First, a comment. There's another common Cincinnati plant that's also called pilewort. So I think it's fine if you mention the scientific name at least once in your talks. After that, you can use a common name. The things I was thinking about is the fact that these plants that I never see any insect damage on any of these plants either. So whether the insects don't eat them, and consequently, I would think the larval stages of insects wouldn't like them, may not like them either. So if it goes into the water, it would not be something that they would eat or digest, or it may even kill. So that made me think that you might do an experiment more directly on the aquatic environment by taking your weak tea, take a tea and dilute it somewhat, and test, pour it into a aquatic environment, into a goldfish bowl filled with some native insect larvae, et cetera, and microbes, and see what happens to that. I would be curious to know if it's actually killing them or they can actually get any nutrient from it.
Yeah, I have actually done that with Daphnia, which is like the fruit fly of aquatic insects. It's the little water flea. I have had students do that. They've done different extracts and looked at how the Daphnia do. And again, it's one of those things, like some species are bad, some are worse. But I'm really interested in tree holes too. So, tree holes are of course where leaves gather and then you have a lot of mosquitoes. So I've wanted to do some of these studies looking at the tree hole communities. It's not just mosquitoes, but there's these great things called rat-tailed maggots and all these other things found in tree holes. And if you change sort of the leaf matter that goes into these tree holes, you can really change the communities. So I'm interested, and I've tried to do that, but believe it or not, when I had a student try and do it, we tried to just get natural inoculation by mosquitoes, and we got like no mosquitoes laying eggs in our water all sitting out for a while. I think we were too late in the season, but yeah, that's a really good question. I have thought about that. And people have done it. I know I've seen talks, I think out of the University of Dayton, where they've looked at different insects and fed them different leaves and then looked at the communities. They had like these flow through feeders kind of thing that they worked with, but I don't remember the results. It was just a presentation I saw at a meeting, but I know people are definitely working on it. And frogs too, frog communities, and sort of changing the basis of the food web for like
And of course, if you
simulated
change
ponds.
one of those creatures, if you change, you lose the insect larvae, you're going to lose other, you're going to lose everything upstream. I mean, everything uphill in the food chain from those things as well.
Right. Yeah. I had a colleague at Penn State. He studied tree holes, and he would manipulate, the food source, which was leaves. And then you saw how the whole community changed from there, just to that base. He looked at like maples versus oaks and saw how, because maples are much more palatable than oaks. So you had a whole different community in maple tree holes versus oak tree holes. Yeah, Deborah, I see your hand is up.
Yeah, thank you. I am wondering, mostly everyone who I talk to about lesser celandine is spraying glyphosate, and I'm very concerned about the use of glyphosate on whatever, the waterways or the ground and other plants and people. And we're actually trying an experiment about using iron chelate, which haven't had a lot of data on it, but we're willing to try something because we don't want to use glyphosate. So I'm wondering if you know anything about any of those matters.
No, I have done experiments looking at control, how to best control ranunculus, and used herbicides. I used, I used Aquaneat, right? So there's some glyphosates that are approved for waterways. The main thing that they found that is toxic about glyphosate is the surfactants that are in them. The surfactants, at least the surfactants, seem to have a bigger impact than the actual herbicide does. So if you use the right surfactant, then glyphosate is not supposedly as deadly to aquatic systems. But I get your concern, because there's a lot we don't know. But when you're dealing with lesser celandine too, it's a little bit high up in the floodplain, so I think you can avoid contamination, and it does break down fairly quickly in the environment. But yeah, it's an issue, right? I mean, I don't know. You've got like one horrible thing or the other horrible thing. It's kind of like a lesser of two evils. If the glyphosate might be a small, short-term impact, if you can control it, but then maybe your woodlot is protected for longer. So it's one of those things. But if you have to like spray the hell out of every year, then you reach a point where you're like, what am I doing?
Well, Tim mentioned about their
Thank you.
treatment of lesser celandine right before the meeting, and they don't have much of it, so they're not spraying much glyphosate on it, which is good. So that's the way to use less glyphosate, is to start treatment early, which is what Western Wildlife Corridor seems to have done, whereas Cincinnati Parks is filled with lesser celandine. At this point, to get rid of it, they would have to use a lot of glyphosate.
Yeah. Well, in the window too. I mean, if you're it's out so early, that if you go out and treat it early, then hopefully there's nothing else you're going to accidentally spray and get with it. You can't get sort of the non-target impacts because it is out really early. And that's one of
There was a,
that's one study I've done, is looked at different time periods. Actually, the earlier that you apply it, as long as it's warm enough, not too cold, the earlier you apply it, the better, because it hasn't, like, once lesser celandine all like forms that mat, it's like lesser celandine on top of lesser celandine on top of lesser celandine. So when you go to spray it, it kills it, but there's a bunch of lesser celandine that you miss because it's under leaves of other lesser celandine. But if you go out early when it's kind of just coming out, you can get it all. It's like all separate clumps, more or less.
there's a paper I ran across several years ago from Ohio State where they looked for the optimum time to spray lesser celandine. And they found a window that depended on degree days, where it's most effective. It's real early isn't as effective, and late isn't effective. But they found this window right in the middle, this sweet spot, where if you sprayed it with glyphosate in that window, it was the most effective
Mm-hmm,
based on degree days.
mm-hmm.
I had a couple other, one quick question, or I have two questions, but one quick question. Is your paper available for free online somewhere, or could you let Tim see it, Tim and several of us see it?
Which papers? That was like six
The paper
papers.
that you were referring to, the one that, or your charts and information from tonight came from, is that from several?
No, I'm in the process of writing that. It's a long
okay.
process.
Okay,
But yeah, no, I presented at several meetings and I've had it nearly written for 2 years, but I need to
Okay.
just finish it. But yeah, when
Okay.
I finish it, I might actually make a goal over spring break to finish it, and then I can share it. Yeah, I just don't have it all written up yet.
my last question is specifically on your plots where you had lesser celandine and you were planting touch me not within that and seeing how it did. The thing that occurs to me is that I saw you're controlling for nutrients and the allelopathy, but you weren't controlling for water. I would think that the lesser celandine would be hogging all the water, allowing less water for the native plants or the plant that you were jewelweed.
Maybe, but it's such a moist habitat. I don't think water was really that limiting.
I do notice that jewelweed in particular, when it's hot or when it's dry, it gets droopy very quickly. If it's too hot or it's been too dry, it gets droopy very quickly. And I would think it would stop growing in height, and maybe the decrease in seed production as well. If it has a period like that, I would assume that even later on in the season, it would have fewer seeds. But so that's just an observation of
Yeah,
mine.
yeah. I mean, you could definitely add water. I already had 3 things, but yeah, you could add another 4th thing to it. But yeah, you could look at water competition too.
That's one more question. It would be nice if you could make something that would poison, that you could water the soil with from garlic mustard. You could water the soil with, and it would kill the honeysuckle bushes.
Right.
That would be a valuable product. I mean, I know you mentioned they're sort of specific about what. I mean, some plants don't mind the stuff as much, and other plants do. But if you could develop products that would hinder honeysuckle, garlic mustard,
garlic mustard,
solution that hurt garlic or honeysuckle and not hurt the good plants, that would be a valuable product.
Yeah, the problem is it's probably going to hurt all the good plants.
Any other questions? There was one item in the chat here. It says, I'm in Northeast Ohio. We have had decent experience with purple loose strife beetles. DOW also uses the beetles in Northwest Ohio.
Yeah, that's sort of a big success story for biocontrol. Those purple loose strife beetles are pretty effective, and they haven't really shifted over to other hosts yet. So they're pretty good biocontrol.
you have any advice for Tim with the new wetland that's near Kirby, our streamside habitat there, as far as controlling these things?
That's not the
big
problem there.
Yeah, the big problem down there is another plant that is real new, that's just coming down the river. And I showed it to Kendra last year when we were over there. So do you have any more information on that one, Kendra?
No, that was the first time I saw it and I don't even remember the name of it. What is it called?
What is it called? It is called, it'll come to me. Trust me.
Japanese chaff flower.
Chai flower, yeah. Right.
Okay.
It's really new within the last couple of years, but it is very bad in that floodplain habitat
Yeah,
because it loves moist soil.
I've never seen that before. That might be like a goat solved. I don't know what it might, maybe animals to eat it. I don't know what it might be, but that whole area is covered with it. So that's a huge issue. And I don't know what it does. It did, does it do anything? I don't know.
It has lots of seeds and it spreads very easily. It spreads mainly first along wetlands, but then it spreads into drier areas afterwards. So it likes water, but it will spread otherwise. So I would say that's a hard one. I saw that
Yeah,
probably
it spreads.
for the first
Right,
time.
I've
maybe
seen it
10
up the
years ago.
slopes. Yeah, I've seen it up the slopes away from the waterways. Of course, it was first introduced in eastern Kentucky and came down the Ohio River. So it was present in the waterway and then spread outward from the Ohio River. But it's spreading out over dry land now.
Okay, last call for questions. No, all right. Well, a couple of closing remarks. First off, Kendra, thank you so much for the wonderful presentation. I loved it and learned a lot. So that's very much appreciated. Thanks to everybody on the phone here for joining us. For those of you who are not members and would like to become members to provide support for our organization, I would encourage you to sign up. You can get information about that on our website. Also, thanks to Kathy McDowell, who leads our events team for Western Wildlife Corridor, for setting this up. Much appreciated. Her efforts there, as well as Marie Finn, who's on the phone here for getting this set up. So that's it. On behalf of Western Wildlife Corridor, thanks everybody for joining us. And I think we can wrap it up here. Take care.
Thank you, everybody.
Thank you.
Bye, everybody.
you.
Great call.
Good to
Bye-bye.
see you.